every single day, just by existing. Cells in your lungs acquire genetic mutations.
Right. It's just a statistical reality of life.
Yeah, exactly. I mean, you take a breath, your cells divide, you're exposed to the environment, and errors just happen in the DNA. It's this constant state of microscopic chaos.
Absolutely.
Yet, despite millions of these errors occurring across the human population, like every second, only an infinitesimally small fraction of those mutant cells ever actually survive, let alone become full-blown tumors.
Which is actually pretty amazing when you think about it.
It really is. And for decades, the scientific community leaned pretty heavily on something called the seed and soil theory of cancer to explain this.
Yeah, which is a very intuitive way to think about it. I mean, the idea being that you have a mutated cell that's your seed, and it wanders around or sits there until it happens to find the perfect, welcoming biochemical environment.
The soil.
Right, the soil. And only when the soil is right can it put down roots and grow.
It kind of implies the cancer cell is somewhat passive early on, just waiting for a lucky break. But today, we are doing a deep dive into a groundbreaking study published in the journal Nature that caught a microscopic heist on camera.
It really is a heist.
It is, because what this research shows is that the tiny mutated seed doesn't passively look for good soil. It actively, aggressively builds its own soil from scratch. It literally hijacks your healthy cells to do the heavy lifting.
It's a massive paradigm-shifting way to look at early disease. We're no longer looking at a rogue cell that just happens to multiply out of control because of a broken brake pedal.
Right.
We are looking at a master architect, a cell that orchestrates the construction of a highly complex, heavily fortified ecosystem right in the middle of healthy tissue.
And the researchers figured this out by tracking the exact step-by-step blueprint of how early lung adenocarcinoma, or LUAD, initiates. And they used a technique called high-resolution single-cell transcriptomics.
Along with spatial tracking in lung tissue?
Yeah, which sounds incredibly dense. But it basically means they weren't just throwing a piece of tissue into a blender and looking at the average DNA, right?
Far from it. Historically, analyzing tissue was a bit like taking a fruit smoothie and trying to guess what fruits went into it based on the taste.
That's a great way to put it.
But single-cell transcriptomics allows us to pull out every individual berry, banana, and apple and read its specific genetic instructions at that exact moment in time.
Wow.
Yeah, they track the RNA of thousands of individual cells in real time to see which genes were being turned on or off as this tumor environment was being built.
Okay, so let's set the stage for where this heist actually takes place. Picture a healthy lung alveolus. These are the tiny, delicate, balloon-like air sacs at the very end of your respiratory tract.
The very deepest part of the lung.
Exactly. This is where the actual magic of breathing happens, right? The exchange of oxygen and carbon dioxide. It's a peaceful, quiet, highly structured environment. The cells there are just doing routine daily maintenance.
Right, just minding their own business.
Minding their own business. And then a critical genetic mutation strikes.
And the specific mutation driving this model is called KRASG12D. It's one of the most notorious drivers of lung cancer.
And the cell it happens to strike is known as an alveolar type 2 cell or an AT2 cell.
Correct.
Okay, so if you're picturing this peaceful balloon structure, what is the AT2 cell's normal day job before the mutation hits?
So in a healthy lung, AT2 cells are essentially the resident stem cells and the local repair crew.
Okay.
The alveolar lining is incredibly thin, it has to be, for gases to pass through it. So if you inhale a pollutant or get a mild viral infection and that delicate lining gets damaged, the AT2 cells step up.
They're the first responders.
Exactly. They divide, they differentiate into other cell types, and they patch the hole. They are the guardians of your lung structural integrity.
So they are built to respond to emergencies.
Yes. But when that KRAS mutation occurs, it fundamentally breaks their software. Well, the classic view of cancer is that a mutation like KRAS just tells the cell to divide wildly and form a mass. But the researchers found something much stranger. The mutant AT2 cell actually enters something called a DATP state.
A DATP state. That stands for damage-associated transient progenitor state, right?
But that's the technical term, yeah. Normally, when a healthy AT2 cell is fixing a wound, it enters this DATP state temporarily. It's a transitional, highly active repair phase.
Okay.
It does the job, then powers down. But the KRAS mutation acts like a jam in the system. It locks the cell in this DATP state permanently.
Oh, wow. I'm picturing a construction foreman on a building site. like a pipe burst, so he hits the panic button, triggering a state of emergency repair.
That's a good visual.
Right. But because his radio was broken in mutation, he never gets the all-clear signal from headquarters. He just keeps shouting into the megaphone, locking the entire site in a permanent, chaotic state of emergency protocol.
That is exactly what's happening. But the foreman needs a way to communicate that panic to the rest of the site. So the mutant cell transforms into a central signaling hub by pumping out massive amounts of a specific protein called amphiregulin, or ERG for short.
So ERG is the megaphone.
Essentially, yes.
Yeah.
ERG is a ligand, which is a chemical messenger. Think of it as a key floating through the extracellular space looking for a very specific receptor, or lock, to bind to.
Got it.
And this ERG signal is a powerful one. It's the starting gun for the entire tumor building process.
Okay, so let's do a quick mini recap for everyone listening before we move to the next step. So bullet 1, AT2 cells are your lung repairmen.
Right.
Bullet 2, the KRAS mutation locks them in that permanent DATP emergency repair state. And bullet 3, they start secreting that ERG emergency signal.
Perfectly summarized.
So my question for you, the listener, is this. Imagine you are a neighboring healthy cell receiving this blaring emergency ERG signal. How would you react to a false alarm that literally never turns off?
It's a great question because the signal has to go somewhere. It has to find its specific lock.
And where does it find it?
Very close by. The ERG signal doesn't travel systemically. It hits the immediate neighborhood first. It goes directly to cells called alveolar fibroblasts.
Fibroblasts. These are these structural support cells, right? The ones that provide the physical scaffolding for the air sacs.
Correct. Normally, these fibroblasts express a marker called PDGFR alpha.
Okay, PDGFR alpha.
Yeah, and in this state, they are supportive, quiet, and helpful. They just maintain the basic framework of the lung tissue. But crucially, on their surface, they have a receptor called EGFR, the epidermal growth factor receptor.
Ah, that's the lock for the ERG key.
You've got it. When the mutant cell's ERG ligand binds to the fibroblast's EGFR lock, it triggers a catastrophic identity shift.
Wow.
By tracking the RNA, the researchers watched this happen. The fibroblasts lose their normal, peaceful PDGFR alpha marker. Instead, they upregulate a whole new suite of genes, most notably one called PDGFR beta, along with genes like ACTO2.
Okay, let's translate those gene names into what they actually do, because just listing acronyms can get overwhelming. When a cell turns on PDGFR beta and ACTA2, what is it actually becoming?
It's turning from a quiet support cell into a hyperactive fibrotic machine. Those genes are essentially the blueprints for building tough scar tissue and contractile muscle-like fibers.
So it changes completely.
Completely. The cell transforms from a supportive alveolar fibroblast into a fibrotic fibroblast.
I think we need a solid visual to lock this mechanism in.
Yeah.
So think of the normal, healthy fibroblast, like a quiet IT guy maintaining a server room. He's just doing routine updates, keeping things running.
Right.
But the moment that ERG emergency signal hits his desk, he completely hulks out. He locks the server room doors, pulls out massive spools of heavy-duty Ethernet cable, and frantically starts weaving a physical barricade around the rogue mutant cell. He's weaponizing his structural abilities.
I love that analogy. And that Ethernet cable he's spinning has a specific name. It's a highly specialized extracellular matrix protein called tenascin C, or TNC. Yeah, and TNC is thick, it's sticky, and it's usually only present in the body during severe tissue injury or early embryonic development.
So it shouldn't be there.
No, it is absolutely not supposed to be hanging around in healthy adult lung tissue.
Yeah.
By forcing the fibroblast to pump out TNC, the mutant cell is essentially wrapping itself in a protective fibrotic cocoon.
Okay, time for another quick recap. Bullet 1. The ERG signal hits the EGFR lock on neighboring fibroblasts. Bullet 2. Those fibroblasts turn into fibrotic fibroblasts, switching their marker to PDGFR beta.
Yep.
And bullet 3, they start secreting those sticky TNC cables to build a cocoon.
Exactly.
So question for you listening. If you could design a drug right now, would you target the mutant cell itself or the sticky web it's forcing the fibroblast to build? Something to keep in mind.
That's the exact puzzle researchers face.
Because, wait, a physical web can't be enough to survive. Your body has an immune system. Eventually, the security guards are going to show up to investigate this unauthorized construction site.
They are.
And the primary guards in the lungs are the alveolar macrophages. And macrophages are literally the Pac-Men of the immune system. Their entire biological purpose is to patrol the tissue, recognize abnormal or damaged cells, and eat them.
Right.
So why on earth do they let this mutant cell get away with this? How does a physical web of tenascin C stop a macrophage?
You're hitting on the core mystery the researchers had to solve. Because you're right, a physical barrier alone wouldn't stop an immune cell. This is where the sheer evolutionary deviousness of the tumor reveals itself.
Oh boy.
Those thick TNC cables aren't just structural concrete, they are active communication lines.
Meaning the web itself is sending a signal.
Yes. TNC proteins bind directly to a receptor on the surface of the alveolar macrophages called TLR4, toll-like receptor 4.
TLRs. Those are the ancient alarm sensors of the immune system, right? They usually detect bacteria or viruses.
They do. But here, the interaction between the TNC cable and the TLR4 sensor totally corrupts the macrophage. Instead of triggering a response to destroy the mutated cell, the TNC protein mimics the biochemical signals of a massive, severe open wound. It tricks the macrophage.
So it lies to it.
Exactly. The immune cell essentially thinks, the tissue is catastrophically torn apart. We need to stand down from fighting pathogens and start aggressively healing this wound.
The ultimate disguise.
Oh.
The tumor is literally dressing up as a trauma.
And the single-cell tracking proved it.
Mm-hmm.
The researchers mapped this exact shift. The macrophages lose their normal defensive profile and upregulate a marker called MSR1.
MSI1.
Yeah. They become reprogrammed. Once in this corrupted state, they stop eating threats entirely. Instead, they begin to rapidly multiply, expanding their population around the fibrotic cocoon, and they start pumping out intense inflammatory signals.
Like what kind of signals?
The primary signal they release is an immune molecule called IL-1 beta.
Okay, let's do our third recap. Bullet 1. Sticky TNC cables bind to TLR4 on the macrophages.
Mm-hmm.
Bullet 2. The macrophages are reprogrammed, turning on that MSR1 marker.
Right.
And bullet 3, they start releasing inflammatory signals like IL-1 beta.
You got it.
So here's a question for the listener. Think about a time you had a chronic injury, like a sprained ankle that stayed swollen and red for weeks. How does it change your perspective to know that tumors literally disguise themselves as wounds that won't heal?
It's a profound shift in how we understand malignancy. It's not fighting the system, it's exploiting the system. And that exploitation brings us to the next phase of construction. Because those reprogrammed macrophages are now shouting inflammatory signals that IL-1 beta into the surrounding lung tissue.
But the immediate neighbors, the first layer of fibroblasts, are already corrupted into that dense fibrotic web. So the IL-1 beta signal must travel further out.
It diffuses into the wider neighborhood. This creates what the researchers call the second wave of the tumor niche.
The second wave.
Yeah. The IL-1 beta hits other normal fibroblasts that are located further away from the central mutant cell. And this signal triggers a completely different kind of reprogramming.
Oh, really?
Yeah. These distant fibroblasts don't turn into the heavy-duty web spinners. They become inflammatory fibroblasts.
So we have two totally distinct layers of corruption here.
Yes. The spatial architecture is incredibly precise. You have a central core, which is the mutant cell wrapped in that tight, sticky, fibrotic web of PDGFR beta fibroblasts.
Right.
Then on the periphery, forming an outer ring, you have the reprogrammed macrophages and these new inflammatory fibroblasts. And these outer ring fibroblasts have completely different markers. They keep their original PDGFR alpha, but they also express a new marker called LCN2.
LCN2. I actually have a special algorithm, like a little mnemonic, to help you listening remember this layout.
Oh, I'd love to hear it.
Okay, think of the letters CFPI.
CFPI.
Yeah. C is for core, F is for fibrotic. That's the mutant cell wrapped in that sticky web. Then P is for periphery, and I is for inflammatory. That's the outer ring of macrophages and those LCN2 fibroblasts.
CFPI. That's a perfect way to visualize it. It's like a terrifyingly perfect bullseye. An inner fibrotic core surrounded by an outer inflammatory ring.
And the researchers didn't just infer this from the genetic data, right? They physically proved it using advanced 3D immunofluorescence.
Yes, they did.
Let's explain how that works, because seeing this must have been a wild moment in the lab.
Oh, it must have been incredible. Immunofluorescence is a technique where you use specific antibodies that bind to the proteins you want to look at, and those antibodies are tagged with fluorescent dyes.
Okay.
So, under a highly specialized microscope, they literally lit up the outer inflammatory LCN2 proteins in one color, say yellow, and the inner fibrotic PDGFR beta proteins in another color, like green.
And there it was.
And there it was. They could visually see this exact concentric bullseye pattern glowing in the lung tissue.
It's physical proof of the blueprint. But wait, there's something backwards about this sequence.
What do you mean?
Well, normally, if I accidentally slice my finger with a kitchen knife, inflammation happens first.
Yeah.
The area gets red, hot, and swollen as the immune system rushes in.
Right.
And then days later, the scar tissue, the fibrosis, forms to seal the wound. But here, it's reversed.
You've spotted the timeline flip. In early cancer initiation, the natural biological sequence is hijacked and completely reversed. The researchers tracked the development over 8 weeks in living mice. The fibrotic core forms first, within the very first week, driven directly by the mutant cell's amphiregulin signal.
Okay.
It's only later, around weeks 2 to 4, that the macrophages expand enough to drive the formation of that outer inflammatory ring.
So the fibrosis is actually driving the inflammation, not the other way around.
Exactly. The tumor secures its physical bunker first and then surrounds it with an inflammatory moat.
So time for recap number 4. Bullet 1, IL-1 beta from the macrophages creates a second type of fibroblast.
Okay.
Bullet 2, these are inflammatory fibroblasts marked by LCN2. And bullet 3, they form that spatial ring we talked about. Remember, CFPI core fibrotic, periphery inflammatory.
Spot on.
So, question for the audience. If you were looking at a tissue sample under a microscope, could you use this special bullseye map to spot a hidden microtumor before it even starts growing out of control?
And the answer is potentially yes. The diagnostic potential is massive. If you see the moat, you know there's a bunker.
Right.
But of course, spotting it is only half the battle.
Which brings us to the most crucial question, really. Now that we have decoded the exact step-by-step blueprint of this corrupted construction site, from the AT2 cell panic to the air egg signal to the sticky web to the inflammatory moat, can we throw a wrench in the gears and dismantle it?
The short answer is yes. And this is undeniably the most hopeful part of the entire study.
I'm ready for some hope.
The researchers discovered that this incredibly complex, fortified chain reaction is entirely reversible in its early stages.
Reversible. Like
Yeah.
not just paused, but actually undone.
Completely dismantled. They proved this using pharmacological interventions. Think back to the very first step we talked about. The AREG signal hitting the EGFR lock on the neighboring fibroblasts.
Right. The megaphone hitting the IT guy.
Yes. When researchers treated the models with a drug called gefitinib, which is a medication that specifically blocks that EGFR lock, the entire tumor niche collapsed.
Oh, wow. So the corrupted foreman is still trapped in his panic state, shouting into the megaphone, but you've essentially cut the wires to the loudspeakers. The neighboring cells can't hear the command to build the web.
Exactly. And because they can't hear it, the fibroblasts lost their fibrotic markers. The sticky tenacin C cables vanished.
They just went away.
Yep. And without the cables to trick them, the macrophages reverted to their normal defensive state. The whole ecosystem just melted away.
That is incredible.
And they saw the exact same ecosystem collapse when they used a highly targeted experimental drug called MRTX-133, which directly inhibits the KRASG12D mutation itself.
Okay, I have to play devil's advocate here for a second. Curing early-stage cancer in genetically engineered mice is a massive scientific achievement, sure.
Sure.
But we've cured cancer in mice 1,000 times over. It is a long way from a mouse lung to a human patient. How confident are we that this exact blueprint operates in us?
That's a very fair point. And the researchers knew they had to bridge that gap, so they moved the evidence up the grading scale. To test this in human tissue without risking patient lives, they utilized human LUAD alveolar organoids.
Okay, for those who might not be familiar, an organoid is basically a 3D mini organ grown in a lab dish, right?
Exactly. They took primary healthy human AT2 cells and cultured them into these complex 3D structures that mimic the function and architecture of a real lung alveolus.
Okay.
Then they used a very clever viral vector system hooked up to a doxycycline switch.
Wait, how does a doxycycline switch work?
It's a genetic engineering tool. They insert the KRAS mutation into the human cells, but it lies completely dormant until exposed to a specific antibiotic doxycycline.
Oh, I see.
So the researchers can literally turn the cancer mutation on just by adding the drug to the Petri dish.
That is wild. So when they flipped the switch on these human mini lungs, what happened?
The human cells did the exact same thing the mouse cells did.
Really?
Yes. They entered that permanent DATP repair state, pumped out massive amounts of the amphiregulin ligand, and initiated fibrotic reprogramming in the surrounding human lung fibroblasts. The mechanism held up perfectly.
But organoids are still lab-grown. Do they look at actual human patients?
They did. They analyzed incredibly detailed single-cell RNA sequencing data from real patients who had been diagnosed with early stage I lung adenocarcinoma.
And the bullseye was there.
The exact same bullseye. They found the mutant DITP-like cells sitting right next to the fibrotic fibroblasts expressing those scar tissue genes, all communicating via the AREG EGFR circuit.
Wow.
Furthermore, they looked at a completely different genetic driver of lung cancer, a mutation called EGFRL858R. And guess what? It builds the exact same niche.
So this isn't just a quirky feature of one specific KRAS mutation.
No, not at all. This ARG EGFR signaling axis appears to be a universally conserved blueprint for how early lung cancers build their homes, regardless of the specific genetic spark that starts the fire.
Okay, final recap time. Bullet 1. Blocking the AR-EGFR pathway dismantles the tumor niche. Bullet 2. Human organoids and real patient tissues prove this happens in us.
Yes.
And bullet 3. It is a shared mechanism across different mutations. So, listener, my question for you is, if this environment is completely reversible in its early stages, how might this change the way we approach preventative medicine for high-risk individuals?
It's the multi-million-dollar question. It moves us away from this archaic idea that we just have to wait for a tumor to get big enough to cut out with surgery or nuke with radiation.
Right.
It opens up the possibility of what we might call ecological intervention.
I really love that phrase, ecological intervention. As we synthesize this whole journey, I'm just struck by the profound, almost terrifying elegance of the biology we've talked about today. Cancer isn't just a rogue, isolated cell dividing in a vacuum.
No, it's not.
It is a rogue architect. It uses your body's natural repair signals, your own structural support cells, your own immune guards, to build a corrupt, fortified city out of your own healthy tissue.
And that reframing is essential for the future of medicine. If we understand the why and the how of the microenvironment, how the seed actively builds its own soil, we open up totally new avenues to heal.
Yeah.
We can move beyond just trying to poison the cancer cells directly, which is, highly toxic to the patient, and start working on uncorrupting the neighborhood. By blocking that one communication wire, that EGFR receptor, the corrupted city falls apart, and the rogue architect is left entirely exposed.
It's an incredible shift in perspective. It makes you realize how deeply interconnected every single cell in our body really is. And to leave you with a final thought to mull over as you go about your day, we've seen today how our own natural wound healing processes are hijacked to build these early tumors. The mutated seeds literally use the biochemical signals of damage to build their soil. So if that's true, it raises a massive question about how we treat our bodies day to day. Could the way we manage every day, chronic inflammation, the small, unhealed damages, the persistent irritants we carry around, fundamentally alter our actual biological risk of developing cancer in the first place? Are we inadvertently providing the building materials for the rogue architect? Let that sink in.